Thank you so much, Mike. That was such an exciting talk and really great to recap over all that research that we've been following closely for a number of years now. So thank you very much. It was enjoyed widely around the world, I think we can say. You mentioned that human interaction is a key factor for improving the welfare of dogs in shelters. And we had some questions coming in from the audience asking whether you think both the dog's individual past experiences, so for example with males and females, and also the quality of the interaction is relevant.
The quality of the interaction that you provide in the shelter?
So I think there might have been some more recent research that just suggests walking down a kennel block and throwing food over the fence is enough to improve.
Oh, well, I mean, there is the study suggesting that walking is really important. You know, and that doesn't surprise me. And that also is another instance of getting away from the harshness of the kennel environment, and it's also another form of human interaction. And, you know, since we found that a passive stranger sitting in the corner worked as long as you were in a quiet place, then I think those two findings are consistent. And I think the walking is a good idea.
And the individual past experiences of the dog, do you think that might tie into that gender effect?
Well, I don't think it has. I don't think it ties into the gender effect because we didn't see. You know, we looked at the gender of the dogs as well, and it, you know, the gender of the dogs didn't make it. It was only the gender of the petter. We don't. You know, at that time we weren't looking at owner releases versus strays, so we can't, you know, categorize dogs in some other way, but it, you know, it worked across dogs enough to produce a consistent, significant effect. So I think, you know, although there may be some effects, but I don't think it just works for a small subset of dogs that come into the shelter.
We also heard that there's a new exploration into the way that maybe inflammation can affect behavior, and we'd love to hear some more about that.
Okay, well, I'd love to tell you about it.
Bring it
Because that's basically what we do with our rodent research largely now. And you know, when you look at a monkey who's been separated for a long period of time, it tends to hunch down like this, and people have always referred to that as despair. When we look at a guinea pig who's simply put into a new infant guinea pig, but put into a new cage for 3 hours, it at first vocalizes like the monkeys do, and then it hunches down like this. Well, you know, it's pretty hard to think that the guinea pig's showing despair. But what it also looks like, if you just looked at that guinea pig, it looks like the guinea pig's sick. You know? And these pro-inflammatory factors can affect the brain to produce behaviors that are called sickness behaviors. And those are associated with a depressive-like mood. Right? And so if we give an anti-inflammatory to the guinea pig, we can reduce the amount that they show these behaviors. You know, suggesting that the action of the inflammation on the brain is actually, you know, one at least key component in what's producing these, you know, depressive-like behaviors.
And what does the cortisol profile look like?
The cortisol seems to, well, it goes up. It goes up. And I have one more point on that is that we're in collaboration with some other people, we're doing a non-human primate study where adult male monkeys are simply brought in from the outdoors to indoor cages. And this is what happens when they're brought in for experiments. And when that happens, if you walk into the room, you don't see much. But if you put a camera in the room and don't have any people in there, you see the monkeys hunching forward. And it seems that, you know, when there's a person there, they're very defensive. And when we looked at measures, cytokine measures and cortisol measures in those monkeys, if we brought them inside and then we looked at the cytokine response before and the cytokine response at the end of 8 days inside, the ability of glucocorticoids to suppress inflammation was reduced after 8 days in the indoor environment. So it seems to be an interaction between the glucocorticoids and the inflammation, resulting ultimately in an increase in inflammation.
I was going to say inflammatory research, but that's going to sound really bad. Research into inflammation seems to be growing a lot sort of in the last 5 to 10 years, and it seems like an area we're going to see grow even further. Is there anything in the dog area that you're aware of happening, looking at inflammation specifically in dogs?
Not specifically, and maybe I missed something. I mean, I know that's something we've talked about, you know, doing. I mean, we're always limited because, you know, the work that we do in shelters, we aim to, it's work to help the dogs in the shelters. We don't want to be invasive in a way that's going to, you know, hurt the dogs in the shelters in some way. But I think I would really love to get a handle on some inflammatory measures in the shelter, and it's something, you know, maybe we'll be able to do that at some point.
It's some of the thermographic cameras and things like that are offering new ways to examine without being intrusive. So it'd be interesting to see where that sort of side of things goes in coming years.
Yeah, often it's the inflammatory mechanisms in the brain that are the important ones, and they're hard to hard to get to.
We had someone on Twitter, Sue, ask if cortisol can suppress the immune system, will a weak immune system also change cortisol levels? So can it work sort of in the opposite direction as well?
System. I can't think of an instance, but I'm not, so to my knowledge, no.
And just looking back at the studies that you conducted, if you could do something again or do something differently, what would you like to redo or add to in your studies?
Besides doing the secluded room earlier?
Yeah.
I don't know, because if I think of that idea, I'll probably try to plan it for the next experiment. So,
I am wondering about, obviously, you're someone who really values both behavioural information as well as the physiological information. It's one of those struggles, I think, with cortisol being recognised as it's not the perfect marker of stress. Mm-hmm. What do you advise, emerging researchers when they're designing their experiments to be really mindful of if they're wanting to capture both a behavioural profile and also physiological profile?
Well, I mean, the first thing I think is to not assume that what you see in behavior is what you're going to see in cortisol, because very often they turn out quite different. And that doesn't mean that one's wrong and the other one's right. But there can be certain stimuli, releasers, if you will, that are producing a certain behavior that doesn't look stressful at the same time that you're seeing a stressful response inside. Certainly I think you really need to be aware of the basics of how the HPA axis works and design a study so that you're really capitalizing on it. And then at the same token, be very aware of the behavioral effects. If you design a study for one and measure both, you're less likely to have successful results. So for somebody new, I would just remind them of what they probably already know, but that.
And even going to more than just one measure physiologically?
Oh,
perhaps?
yeah, certainly. If cortisol is the best measure, I would say, other people may argue, but is the best measure for these short-term psychological stressors, maybe you won't get as response with another system. But the more information you get, the better you can characterize it. And if you do get some other indication that that complements, that supports it in some other way, then you can be much more comfortable that the cortisol isn't going up because it's youth stress, something that's an activity, but arousal, but not a distressful event.
I guess oxytocin's been getting a lot of media lately,
Mm-hmm.
and it's become a bit of a sexy hormone in our field.
Mm-hmm.
Not as in it's actually a sexy hormone, it's an attachment hormone. But have you been following the oxytocin research? Are you finding that interesting?
Sure. The oxytocin research has actually been going on in basic research for some time. And I think it really is interesting, and it can potentially reduce the HPA axis. I think we have to be very cautious because, it's a very easy one to jump on the bandwagon for. It's the love hormone, and it may not always be that. It has a lot of positive effects, and I think it's a really interesting one. And I think, it may very well be involved in what we're looking at with the dogs. That's something I would like to be able to do too. But, but as long as you show me the data, you really have to be comfortable that the effect you're seeing is really due to the oxytocin, and not just assume that if you're seeing some sort of positive affectionate effect, that is definitely due to the oxytocin, because you don't know that that's the case.
Well, I think something we've seen a little bit just on social media is people get very excited and they say, hey, spray some oxytocin up that dog's nose and they will feel better. So any thoughts on that?
I've thought about it. No, if you spray oxytocin up the nose, this is one way that peptides can actually get into the brain. And it has been done for people, for instance. And if I remember this finding right, but if you have a stressful situation where somebody tells you have to give a speech in 5 minutes and you're going to do it in front of this board of people wearing lab coats, your cortisol levels go up. If somebody sprays oxytocin in your nose, your cortisol levels don't go up as much. All right. And if you get social support, you have a best friend there, they don't go up as much. And if you get both of them, they tend to go up a little bit less than in either case. So it could very well be.
Well, we did that before we came out today, so we're feeling pretty great. No, I'm totally kidding. I have a friend, so we're good.
And we've got the audience taking off their lab coats. Excellent. All right. Well, I think we might leave that with everyone for now. What a way to end. And we'll come back to you, I guess, with the rest of the panel later on this afternoon. So thank you so much for your talk and for this opportunity to ask questions.
All right, thank you.
Thanks.
Thanks, everyone. We will be back shortly, very shortly, with our next speaker.
Okay. Okay.